What is the osmotic pressure? It is the minimum pressure required to prevent the passage of pure water. Pure water generally has a tendency to enter the cell. So as it tries to enter the cell, there will be a pressure to prevent it from entering. That is what is called osmotic pressure. It is when the two solutions are separated by a semi-permeable membrane, they're having different concentration. So that way the osmosis will take place. But how long the osmosis can take place? How long it can take place? Theoretically, until and unless both the solutions which are separated by a semi-permeable membrane have equal concentration. But practically, it doesn't happen. As we can see in that experiment of the thistle funnel, water will go on entering into the thistle funnel. But how long? Because as the level of the water column rises here, it will also have its own weight. So water is trying to enter and the weight of the water is being exerted downwards. So though there will be a difference in concentration, but after some time, the weight of the water column will be so much, it will stop. Now let us see the different types of concentration of solutions. This is generally compared with the concentration of the cell sap. This is known as tonicity. It is the relative concentration of the solution that determines tonicity is the relative concentration of the solution that determines the inward or outward direction of diffusion, whether the water molecules will diffuse out of the cell or diffuse into the cell. That depends on the concentration of the solution which is around the cell, and its concentration of that solution which is outside is compared with that of the concentration of the solution inside the cell. Now, based on that, there are three types of solutions: hypotonic, isotonic, and hypertonic. In the hypotonic solution, the solution outside of the cells, which has a lower concentration of the solutes as compared to the solution inside the cell. So it has a lower concentration. The solution which is outside has a lower concentration. So lower concentration means it has more water molecules compared to the fluid which is there inside the cell, or the solution which is there inside the cell. So such solutions are called hypotonic solution. Next we have the isotonic solution. In the isotonic solution, we have same concentration. The concentration of water molecules and the solute on the both the sides of the cell is same. Means the concentration of the solution outside the cell and the concentration of the solution inside the cell, both are same. Now, if you see hypertonic solution, the solution has a higher solute concentration. That means it has less water. So the concentration of the solution is higher, which is outside the cell, compared to the fluid inside the cell. So it has less water. Now, based on that, When a cell is placed in such solution, we see certain phenomena taking place. So three types of phenomena are there: turgidity, flaccidity, and plasmolysis. So what is turgidity? When a cell is kept in water, just plain water, if you keep a cell, or a solution which is having higher concentration of water molecules compared to the cell sap, in such cases what we see, endosmosis taking place. So water will enter the cell, it will go on entering. A time will come that the cell cannot accommodate more water. It will not be able to accommodate more water. So in such a state, the cell is completely filled up with water, and we call this condition turgid. We call this condition turgid when the cell has reached the maximum level of absorbing water. The cell swells up. So this phenomena is known as turgidity. Now the opposite thing can happen when you keep a cell in a concentrated solution. Concentrated solution means there is less concentration of water molecules in the solution compared to the cell sap. So in that case, what happens is exosmosis will take place because water molecule is more inside the cell and it is less outside the cell. So water molecules from the cell will move out. As the water molecules come out of the cell will now become soft and limp because it's losing water. It will not have that stronger turgid system. Instead, it will become very soft and limp. It no longer remain strong. And so this condition is known as flaccid. And this phenomena is called flaccidity. Next is plasmolysis. Now, if you put a cell in a hypertonic solution is a very highly concentrated solution where it has minimum water. So here exosmosis will take place, no doubt. Water will go on coming out from the cell by exosmosis. The cell will start losing water continuously. And what will happen? So much water will be lost that the cytoplasm inside the cell will shrink. The cell membrane also will withdraw from the cell wall. And this shrinkage of the protoplasm, that is cytoplasm and cell membrane together and other contents of the cell, which will shrink and move away from the cell wall. Such phenomena or such condition is known as plasmolysis. This generally happens when there is excessive loss of water by exosmosis. So it is known as plasmolysis. So we can see here the picture when we put a solution in hypertonic solution. So water goes out and all the contents of the cell, they shrink in the middle. That is plasmolysis. And when you put in isotonic solution, entry of water and exit of water is same. So there is no change in the shape of the cell. And if we put in a hypotonic solution where the concentration of water molecule is more, so water will enter the cell and the cell will get swollen up and it will become turgid. So hypertonic causes plasmolysis. Isotonic does not change. It will still remain flaccid. And in hypotonic solution, the cell will gain water, by endosmosis and it will become turgid. Now let's go to another phenomena called deplasmolysis. Now, as we have seen in case of plasmolysis, the cell has become uh, plasmolysed when you put in a hypertonic solution. Now, if the plasmolysed condition of the cell continues for a longer period of time, then the cell may not be able to gain water anymore and the cell will die. So, to get the cell back to its normal state, what we have to do, the cell should be kept in a normal solution of water or normal water. So when we put in normal water, water will re-enter the cell and the cell will regain its original position. So this phenomena is known as deplasmolysis. So deplasmolysis means it is the regaining of the normal condition of a plasmolysed cell. So when a normal condition is attained, when you put a plasmolysed cell in plain water, that is known as deplasmolysis. Now, when a cell is in turgid condition, when water enters the cell, we can see there are pressures exerted in the cell. Because of water entering, the cell contents swell up and they exert pressure on the wall of the cell. So that is called turgor pressure. There is the inward pressure. The inner contents, so the inner contents exerting pressure and they are pressing against the walls of the cell. That is called turgor pressure. And wall pressure, to balance the turgor pressure, wall also exerts or gives certain pressure. If the wall doesn't offer some pressure, the contents will exert so much pressure that the cell may burst. So to balance the turgor pressure, the cell exerts some inward pressure. That is what we call the wall pressure. In a turgid condition, the turgor pressure and the wall pressure, they balances each other. But if a time comes when the wall pressure cannot withstand the turgor pressure, in such condition, the cell will burst. Now, this turgid condition of the cell or the turgidity in the cells of the plants, what are it? What are the uses or usefulness to the plants? So this helps in providing rigidity to the soft tissues of the leaves. As we know, plants don't have any skeleton to support itself. So it is the turgid condition of the cell. When the cells get filled up with water, they become turgid. They provide strength, mechanical strength to the soft tissues in the leaves. So leaves can remain wide open. It offers turgor pressure that helps the seedlings to push through the soil. We can see they come out of the soil as the embryo grows. Even this helps the roots to crack the walls. You can see plants growing in old buildings and the roots have cracked the walls, passing through those cracks. Thirdly, the turgor pressure in the root cells, they build up root pressure because of continuous absorption of water. As the cells become turgid, they help the water to rise up through the xylem, that is called root pressure. Fourthly, turgidity in the guard cells, they help the guard cells to open and close. We can also see such kind of turgor movements in the sensitive plants of Mimosa pudica, touch me not plant. When we touch the leaves of the plant, we see them closing. So when we touch the cells at the base of the stalk, they lose their turgidity. That is why they close. But after some time, they again gain the turgidity and the leaves open. So this kind of turgor movement is also caused by turgidity. Now let's see the application of plasmolysis. So as we have seen in case of plasmolysis, the cells they lose water, and the water comes out of the cell protoplasm shrinks. What application we can do in our day-to-day life? So plasmolysis, this phenomena can be used to kill the grasses or weeds, unwanted plants in our lawn or playground. So we can sprinkle salt around the base of the plants. So as the salt comes in contact with the soil water, they dissolve and the soil water becomes hypertonic. So the root hair cells are now, as they're surrounded by a hypertonic solution, the root hair cells has to lose water. They lose so much water that the cells become plasmolysed. As the root hair cells get plasmolysed, they cannot take in water. So if it continues for a longer period of time, the cells die. So the root hair cells die, the plants also get killed. This phenomena is applied for salting of meat, then making of pickles, because these fruit pieces or the meat pieces are kept in very strong hypertonic sugar or salt solution. So as you keep them, so water from inside the cells get drawn out, they get plasmolysed. If any bacterial cell or fungal cells are there, they also get plasmolysed, they also get killed. So there is no chance of spoilage of the pickles and the meat. Thirdly, this has to be thought when the farmers apply fertilizers in the agricultural fields. So after application of fertilizer in the fields, the farmer should water the fields, or before application, water should be there in the field. Otherwise, the soil water will become hypertonic, and that will lead to the plasmolysis of the root hair cells of the crop plants. So if it continues for a longer time, all the crop plants will die. So farmers need to water the fields properly after adding fertilizers. If you get a sprain while playing, you find that your ankle swells up. To relieve the pain and the swelling, what the doctors advise, to put our leg in salt water, warm salt water. So when we go on dipping in the salt water, as it is a concentrated solution, so the fluid from that swollen area comes out. The cells lose the water. The swelling is generally because of accumulation of fluids. So the fluids are lost by exosmosis. The cells get plasmolysed in that area, and so the swelling subsides. Pain also subsides. So these are some of the applications of plasmolysis. So what is the significance of plasmolysis? Plasmolysis explains the process of osmosis. Plasmolysis taking place means water is moving out from inside the cell to the outside. That means exosmosis is taking place. So it is showing osmosis can take place. It also provides permeability of the cell wall. That means the cell wall is permeable, it shows, and also shows that the cell membrane is permeability. That is why only the water molecules are lost, not the solute molecules, not the minerals inside the cell. It also shows the cell is living or dead. So if a cell undergoes plasmolysis, means the cell is living. That means water has come out of the cell by osmosis. Osmosis can only take place across a semi-permeable membrane. So when the cell membrane is living, then only it can remain semi-permeable. So as long as it is semi-permeable means the cell is a living one. So that proves that the cell is living. If plasmolysis does not take place, that means the cell is dead. It also determines the osmotic pressure. It will show the direction of water entering or leaving the cell. Now we come to another phenomena of absorption of water, that is the utilization of energy. As we have said, this is a active process. We call it active transport. So in the active transport, substances like salt or ions, they move from a region of lower concentration to a region of higher concentration by utilizing the metabolic energy. Through metabolism, ATP is generated. So they utilize the ATP and take in substances from their lower concentration into the cell where their concentration is high. Because the root hair cells or the cells of the plant body, they are continuously in need of the ions and salts, the minerals. They have to absorb them from the soil because these are the building blocks of certain compounds inside the plant body. So by utilization of energy, those substances are taken from the soil along with the water. So here they have to utilize because the concentration of those ions and salts are very less in the soil compared to the ones already they have inside their cell. Their concentration is high, but still the plant cells need them. So they have to be taken. In that case, the energy is utilized. There are some carrier proteins in the cell membrane. The cell membrane is a living one. So carrier proteins to carry those ions from outside the cell to into the inside the cell, they need energy. So ATP is utilized and this carrier protein carry those ions and salts from the soil water into the cell sap by utilization of the energy ATP. So it is a direction which is generally opposite to the process of diffusion. Diffusion takes place from higher to lower, and here it is taking place from lower concentration to higher concentration. So these are the different processes we have already discussed. Now, as we have said, that is the role of this one, as I've explained. So here you can see, in short, it's written, active transport serves mainly to maintain the normal balance of the ions in the cells. So to obtain the ions like sodium and potassium, these ions are forcibly carried inward from the region of their lower concentration to the region of their higher concentration, that is inside the cells. The energy required for this is supplied by the cells in the form of ATPs. The chief nutrients carried by active transport are nitrates, sulfates, potassium, zinc, manganese, and sodium. Normally, these nutrients cannot pass through the cell membrane of the root cells easily. So for that, they have to be taken in by utilization of the energy ATP. Now let's come to the ascent of sap. What is ascent of sap? Water has entered into the root hair cells. Now, how will this water from the root hair cells, from the roots, go up to the leaves? Because this water is needed in the leaves. Water is one of the raw material for the process of photosynthesis, and photosynthesis takes place in the leaves. So water has to be transported from the roots to the leaves. So the process of ascent of sap, it is the movement of the water and minerals from the root to the leaves through the xylem tissue. It is through the xylem tissue, water and the minerals which are absorbed by the roots are carried from the roots to the leaves through the xylem. This whole phenomena or process is known as ascent of sap. Now, what are the forces responsible for ascent of sap? Capillarity, root pressure, adhesive force, and transpiration pull. It's not a single process which can undertake the ascent of sap. It's a combination of many forces which ultimately helps in the rising of the water from the roots to the leaves. Let us see one by one. What is capillarity? Capillarity is the movement of water along the narrow diameter of the xylem. Xylem is like tubes. The xylem vessels are like tubes, and the cells are hardly seen. Cells are generally very much small, microscopic in nature, and the diameter of those xylem vessels will be so much narrow. Because of this narrowness or very small diameter or uh, thin diameter of the xylem vessels, they help in the movement of water from lower level so that it can rise up to fill up the vacuum caused due to the loss of water by transpiration from the leaves. So as the water is generally lost from the leaves, that creates a partial vacuum in the xylem vessels. So this narrow diameter of the xylem vessels that help in the rising of the water molecules from the lower level to the higher level. This movement of water from a lower level to the higher level through a very narrow diameter of the xylem vessels is known as capillarity. So this is generally because as the water vapor is lost from the upper parts of the, so because of the vacuum, there it creates a force, a pull, which is pulling the water to rise up through the narrow diameter of the xylem tissue. So narrower the diameter, the more it is narrow, more will be the rise of the water by this capillary force. So plants use this capillary action to bring up the water from the roots to the uppermost part of the plants. So these molecules and molecules, they generally attract each other. There is force of cohesion, force of adhesion. So all these come to play, and that creates a suction force. That is how water rises up through the xylem. The next factor is, or the next force which is responsible for ascent of sap, is the root pressure. This root pressure is developed in the roots. That is because of continuous influx of water, means continuous entry of water by osmosis, diffusion, whatever it may be, and that is causing the cells to become turgid and flaccid. There's alternate turgidity and flaccidity. As the water enters the cells, it becomes turgid. As the water goes out of the cell, it becomes flaccid. So this alternate turgidity and flaccidity is continuously happening in the roots of the cortex. This is what we call root cortex. That is in the root hair cells as well as in the root hair cells. So that is creating some force which is pushing the water to rise up. That is how water rises up through the xylem. A force is created in the cortical cells of the root that pushes the water to rise up through the xylem. That is what we call the root pressure. So because of the root pressure also, it helps in the rising of the water through the stem into the leaves. The next forces which are responsible is also the cohesive and adhesive force. Cohesive force is between the same type of molecules. So here between the water molecules, one water molecule will attract another water molecule, that is by the force of cohesion. As one water molecule rises up, they pull the next water molecule from below to also rise up. So a continuous column of water is produced through the xylem. Besides this, there is another force called the adhesive force. It is the force between the water and the xylem. And because of that, the water clings, or it is not allowed to come down because of the force of gravity. It remains attached to the xylem. And that adhesive force also helps the water to rise up to the topmost part of the plant. Then we have the transpiration pool. This is another one. So this is a suction force, as we already discussed before also, is a suction force created because of continuous loss of water from the upper parts of the plant. Because of transpiration, water is lost, a vacuum is created, and that creates a suction force. So a deficit, a great water deficit takes place in the cells of the leaves. And to fill up that deficit, to fill up that vacuum, water is drawn osmotically from the xylem cells into the leaf veins. This from the by the surrounding veins, because veins are also there in the leaves. From there, water is being pulled into the cells of the leaves. And because of that, this suction force which is created in the uppermost part of the xylem cells, that helps the water to rise up. That is what this suction force, which is pulling the water level to go up, is known as the transpiration pull. So it is not a single force which causes the ascent of sap. It is a collective force, is a combined forces of transpiration pool, cohesive force, adhesive force, capillarity, all they work together, root pressure, all they work together to help the water to rise up through the xylem. So that is all from this part of the chapter. So my request will be to all the students of class 10, please go through the exercises at the back of the chapter, practice them. If you have any problem, you can come in contact with me. My WhatsApp number is there in the assignments, the pages of the assignments which we have uploaded in our school site. There my WhatsApp number is there. You can WhatsApp me to have any clarifications about the questions and answers which are there at the back of the chapter. The latter part, I maybe I will post some questions and answers as in the school site. Please continuously visit the school site spsagartala.ac.in. You have to come in contact with those sites. You visit them every now and then to check up any updates coming up there. I hope you will be studying not only biology but other chapters, other subjects. Please remain at home, be safe. Thank you. That's all for this chapter, absorption of roots. I'll be coming back with another chapter in a few days from the second term syllabus. Thank you.